Hello, everyone. Welcome to today's webinar, Lost in Translation, Moving Your Research to Clinical Trials, presented by STEMCELL Technologies. This is the first webinar in STEMCELL's Cell Therapy webinar series. My name is Monica Tang, your moderator for today, and I am the Senior Program Manager in the Services for Cell Therapy program at STEMCELL Technologies. STEMCELL is a global leader in supporting life science research by developing specialized cell culture media, cell separation products, and also accessory reagents. We have a really wonderful panel of speakers for you today. Dr. David McKenna is a professor in laboratory medicine and pathology at the University of Minnesota. He holds the American Red Cross Chair in Transfusion Medicine, and he serves as the Director of the Division of Transfusion Medicine and Medical Director of Molecular and Cellular Therapeutics, the university's cGMP facility. Dr. Adrian G. is Director of the GMP Cell Processing and Vector Production Core Facilities at the Center for Cell and Gene Therapy at Baylor College of Medicine. His research interests include cellular therapy, stem cell transplantation, and regulatory issues and quality assurance. He is a co-founder of the International Society for Cell and Gene Therapy. For today's agenda, Dave will be presenting first on scale-up and optimization of cell therapy manufacturing. Adrian will then follow with his presentation on the development of clinical scale manufacturing. There will then be a Q&A session at the end. I'm now going to hand you over to Dave to begin the presentation.
Thanks, Monica. And thank you to STEMCELL Technologies for the invitation to be part of this webinar today. As Monica mentioned, I'm going to discuss scale-up and optimization of cell therapy manufacturing. These topics are huge. I mean, you could have a full several-day meeting on these. So we'll hopefully touch on some things of interest and generate some good discussion. I have no disclosures for this particular topic, although towards the end of the presentation, I'm going to touch on some actual examples, and those are related to trials that were funded in part by NIH, specifically PACT from Heart, Lung, and Blood Institute and the Department of Defense. All right, these were the webinar objectives that you've all seen in the webinar description. I'm going to focus on these couple here, the product development process from proposal to clinical use, and then I'll touch on kind of the ideal set of team members to ensure success. This is our facility in St. Paul on the University of Minnesota St. Paul campus. It's Molecular and Cellular Therapeutics. It's a standalone cGMP facility, roughly 36,000 square feet, and the cell therapy group is in this building, one of the bigger groups, and takes up roughly half of the facility with labs and QC space, et cetera. All right, I always like to kind of paint a little backdrop for talks like this, just to kind of discuss a little bit about translational research. I think the term itself is becoming more clearly defined, but I think over the years it was less so. We all understood what basic research is and what clinical research is, and we just kind of lumped translational research in between there as kind of that the bridge between the bench and the bedside. I think as time has progressed, we've gotten a better understanding, and maybe in part the term was less clearly defined because it is quite broad and has many different aspects. One definition that I like is a bit old now. This was from a CTSA announcement back in 2007 from the NIH, where they defined translational research as the process of applying discoveries generated during research in laboratory and preclinical studies to the development of trials and studies in humans. This figure I often use in talks when I'm discussing translation. I think it nicely characterizes the broad array of skill sets needed to move something from the bench to the bedside. You can see on the far left working with basic scientists and ultimately going into the clinic working with clinical scientists, and all the people kind of in between. In the lab, we get, we get involved usually before tech transfer, but that's, a very tangible area where we're involved. And then certainly clinical manufacturing, and then there's that, that area in between, all that scale up and optimization. And all of these aspects of translation that involve the product, again, are going to involve a variety of people. It takes a small village, essentially. So just for the product, you're needing to involve these people and probably more. Probably leaving people out. I apologize to anyone out there that with a different skill set. But you would need to include the lab and medical director, the lab and facility leadership, research and development team, quality assurance team, regulatory folks, finance and contracts people, especially if you're working with outside groups, materials, people, and certainly the principal investigator and other scientists involved. And over the years, this is our product development process, and over the years, this has been honed in large part through our involvement with that NIH contract I mentioned, the PACT, or Production Assistance for Cellular Therapies. Adrian's group at Baylor and our group here at Minnesota have been part of that contract since 2003. And for us, certainly, it's helped push us along and helped us to hone our skills in moving products from the bench to the bedside. And so this PDP, or product development process here, was really spearheaded by Diane Cadillo, who's our facility operations director and technical supervisor in the lab. And you can see again here under each of these 5 steps in the process, just a variety of activities requiring different sets of skills. So again, it's not a single person that can do this for sure. And I'm more of a lumper than a splitter, so I kind of move that 5-step process into 3 tiers. And kind of tier 1 is where we're interfacing with the research lab. That would be the proposal and assessment from that previous slide. And then the tier 2 is where we're kind of bringing that technology into our house in the Molecular and Cellular Therapeutics Facility, or MCT. That would be the development stage, and that's going to be one of the bigger, more time-involved stages, certainly. And then ultimately, when you move to qualification or validation of your manufacturing methods, and then into the clinical manufacturing realm, and that would be corresponding to the validation and clinical use from that previous slide. So this is optimization in one slide. These are just some of the considerations, kind of the big picture. Certainly, if you can get reagents and supplies all made under current good manufacturing practices, you would do that. Sometimes you can't. You're looking at instrumentation and culture vessels, trying to optimally have closed systems, automation if that's available, and looking at kind of the scale up and scale out. And I'll talk about that in the next couple slides. The manufacturing plan will get a lot of attention, and logistics, things like media changes. Maybe in the research lab, the media was changed too often or too little. Is your final product fresh or frozen? That's going to have implications on several fronts, but certainly a frozen product may allow you more flexibility in your patient calendar, for example. If you're using a fresh product, you need to time that. If it's a 2-week culture, you need to have that timed right so that the patient is able to get the product when they need it. And often these types of products involve patients, being admitted and maybe having a preparative regimen of some sort. So it gets quite complex, working with the nurse coordinators and the clinical team, et cetera. The scale of activities, again, you're going to be looking, maybe moving from wells to bags or other types of culture containers, and moving from treating mice to people. The cost, we always want to contain, and then you're going to be looking at product testing. There's certain tests that are often, with all cell therapies, things like viability and sterility, immunophenotype, endotoxin, et cetera. But then there's some unique to different product types, especially your functional or potency assay. And also you'd start to plan for your stability testing, and that may be commensurate with the phase of your clinical trial. So if it's a phase 1 clinical trial, it may not be that involved of a stability plan, but you certainly have to initiate one and meet the expectations of the FDA. You're going to want to focus on timelines and milestones. And I always like to say that, you know, you don't always meet your timelines and milestones when you ideally wanted to, but if you don't set them, you certainly won't. So a quick word or two on scale up. This is my attempt at kind of illustrating scale up. You first need to decide if it's necessary. It often is. I would say most of the time it is, because often in research labs, the scientists will be working with wells, and we need to get into ideally closed systems, or if not, you know, maybe instead of working with 100 T-flasks, work with 10 cell factories. That'll decrease the likelihood of microbial contamination and make the whole workflow better. Is the process scalable? Usually they are, and you're targeting something that will deliver an equivalent product or ideally a better product. And again, cost, or the goal here is to decrease cost. Scale out might be suitable for some products. Certainly a third-party off-the-shelf banking strategy might lend itself to scaling out. Most of what we do in the academic setting is, you know, scaled up and not necessarily scaled out. But it might be the right thing to do in certain situations. Again, if it's like a third-party off-the-shelf bank where you can manufacture 300 doses and freeze them down. Cost can be high, but it's really a math. It's really just math. I mean, sometimes it actually is the most cost-effective way. So a big part of that optimization will be those engineering runs where you're looking at maybe the whole manufacturing process, or maybe just some steps in the manufacturing process. It really depends on the level of experience with that particular technology. Usually this will involve multiple arms to test different reagents or vessels or other aspects of the culture. Usually these are small scale earlier on, big enough to, you know, to accommodate some of those arms for the variables, but you're really not doing full-scale clinical runs until you're more settled into your methods once you've decided, you know, those critical variables. Then you would finalize your standard operating procedures and batch production records and develop a qualification or validation plan and then execute it. And typically in cell therapy, that might be 3 successful, meaning meeting pre-established specifications, which are often lot release criteria. Once you've done that, you would write your chemistry, manufacturing, and control section and submit that with your IND. And then in 30 days, you'll hear from the FDA. And if not on clinical hold, you would be ready to move forward. So this is where I was going to jump into the examples real quickly. So we've been working for a few years on MSCs, mesenchymal stromal cells, in acute lung injury, supporting phase 1, phase 2A, and now we're in a phase 2B trial. This is just from the phase 2A trial. Like most MSCs, or maybe all trials, it was shown to be safe. But one of the conclusions was, you know, unfortunately, larger trials are needed to assess efficacy. One of the biomarkers that was tested in this trial that was led by Dr. Michael Mathey at UC San Francisco was angiopoietin-2, which is a you know, part of a member of the family of vascular growth factors, which actually, in this case, promotes cell death and disrupts vascularization. So he noted that there was a drop in angiopoietin-2 with the patients that received the MSCs. And this is shown here in red, the MSC patients versus the control group, the placebo group. Viability was also widely ranging in this study, and was thought to perhaps be due to washing. There were 5 sites, and it was standardized as much as possible, but the washing was thought maybe to have beaten up the cells on thaw. And there was a post hoc analysis actually then that showed that dividing the viability up into tertiles, there was a viability-dependent effect on the angiopoietin-2 concentration in the plasma. So the patients receiving or the patients with the more viable cells had a greater drop in the angiopoietin-2. And looking back, we tried to see what the effect might have been on the cells with washing and reconstitution compared to just thawing and diluting, and it was there was a clear difference. It did seem that spinning the cells down certainly kind of dropped the viability. So the issues with cryopreservation of MSCs is widely known. There are many papers describing the impaired function post-thaw, including from Jacques Gallipo's group in 2012, where there was reduced immunomodulatory activity. Others have shown a drop in homing ability and disruption in actin cytoskeleton. So for many, the solution is to, after thaw, to reculture the cells. The issue with this is cost, and it also means you need to do additional testing, including lot release testing. But maybe greater than that is the lack of access of these cell therapies then to places that don't have a advanced cell therapy lab. So we need to come up with better cryopreservation, I guess, is my thought there. At the same time, we were looking at the impact of cryopreservation, and we at the time made our MSCs from both fresh and frozen marrow. So we looked and tried to see what's the difference, depending on your starting material. And the growth characteristics were similar, whether you use fresh or frozen bone marrow. However, the viability was better if you used fresh marrow. So we decided we needed to work on a new method, and the FDA, moving forward with the phase 2B study, suggested that we, rather than making MSCs from both fresh and frozen marrow, that we needed to decide on one approach, either make it with fresh marrow or with frozen marrow. And so we decided to make a consistent product using fresh marrow, but we would go to passage 1 and freeze down the MSCs at that point, and then thaw a vial at passage 1 and carry it into passage 2, harvest it, and then freeze as the final product for patient use. And so these were a few things that we looked at for manufacturing improvements. I thought they would be a good example of kind of, showing optimization in real life. So again, in the bottom left, you can see that whether you used the vials at P1 that were frozen or whether you used fresh marrow from that same collection, they were relatively equivalent and showed expansion that was within our historical range of fresh MSCs or fresh MSCs made from fresh marrow. And then the upper right and the bottom right, we decided to look at an instrument to harvest the cells more gently. And this is using the LOVO, which uses a spinning membrane to wash the cells, as opposed to a centrifuge-based technology like the COBE 2991, where cells that enter in the beginning of the harvest may be in that centrifuge for an hour or more. And so we were looking at recovery and viability, and as you can see, the LOVO performed as well or better than our current, or at that time, current method using the 2991. And finally, we looked at the cryopreservation, trying to optimize the cryo solution. This is one set of experiments looking at a couple different DMSO concentrations. Our in-house, or our method we used at the time, was blue, the one on the far left, that lighter blue, 10% DMSO and plasmolite and human serum albumin. All of these are relatively decent for viability. If you look on the left side, the scale is, pretty high. But in the end, we ended up repeating a few more experiments, looking at some lower DMSO concentrations, and ultimately chose a commercial product for a few reasons, as outlined there. The cryo solution would be manufactured under CGMP. Companies often will have a master file, and in the couple cases from the groups we went with, they had their packaging of their cryo solution was optimal, meaning in bags and at good volumes that would be needed for clinical manufacturing. We have several other studies ongoing to optimize cryopreservation with MSCs, and we're working with one of the engineers at the University of Minnesota here, and she has a new company, Blue Cube Bio, where they really focus on replacing DMSO and using sugars, sugar alcohols, and amino acids to cryopreserve cells. And then there's a few other things we're working on with the School of Engineering, different agarose, freezing approaches, etc. But I really do think that there needs to be increased attention to cell-specific cryopreservation, and I think that's kind of the direction we're heading with many of our cell therapies. So I think that's a wrap up for my talk. So in conclusion, successful product development does require a team with a broad array of skill sets, and an established product development process is essential for smooth translation from the bench to the bedside. And I included kind of a typical life cycle of a cell therapy product here, just to show that, even as you deliver products to a patient, you're always, trying to look and see how can we improve this, whether it's improvement in collection, shipping, cell processing, receipt and storage, or delivery to the patient. With that, I want to thank you for taking the time to listen to this talk. I want to thank the group in our cell therapy lab listed there, especially Diane Cadillo, Darren Sumstead, who are involved in a lot of the work that I discussed there. And with that, I want to again thank you and wish you a happy St. Patrick's Day.
Thank you, Dave. That was really great. I especially enjoyed the St. Patrick's Day motif through some of your slides. Next up, I will pass it on to Adrian to begin his presentation.
Yes, hello everybody. I hope you can all hear me. I'm going to supplement what Dave has said by talking a little bit about some other issues with clinical scale manufacturing and how you develop it. What I'm going to talk about is the choice of culture systems, whether to use automated or manual systems, and some other considerations that have to be taken into account when developing these kinds of procedures. I'm then going to give you an example of trying to reduce costs and timelines, and then just conclude with a little bit of information about possible future directions. The choice of culture system is very dependent on a number of issues. For example, obviously the number of cells required, and that will depend on the dose and the number of doses that have to be given. And then the number of patients on an allogeneic cell therapy trial. So if you're making allogeneic cells, you will have to consider the total number of patients that will accrue and the cell dose and the number of doses. You're then going to have to determine whether you need a system for culturing adherent or non-adherent cells. And if you're going to culture adherent cells, what is the best choice of carrier to use for them to attach to? You're also going to have to consider the manufacturing time available. In some cases, cell therapy products are required at relatively short notice. And are you going to be able to come up with a manufacturing system that will address this? Then you'll have to consider whether there is an available closed system or whether you have to use an open system for culturing, whether it's going to be a manual or an automated system, and then finally whether the product is going to be administered fresh or cryopreserved. And obviously, cryopreservation is a much easier method since it gives time to do all of the release testing example. But for fresh cells, obviously you have to have everything timed extremely closely so that the cells are available at the time the patient needs them. We're going to address now the issue of scale-up, and Dave has talked about this. And I think there are really two choices. Do you use more of the same culture vessels that we used at the research level? So if you were growing cells in a T75 flask, do you just use more T75 flasks, or do you go up to a T225 flask? Or do you use a different kind of approach for producing larger cell numbers? This is the method that we were using many years ago for growing cells, mesenchymal stromal cells, for treatment of patients with stroke. And you can see that we started off with a relatively small volume of bone marrow and then progressively split it until we ended up with incubators full of flasks to produce the cell numbers that were required for the trial. And obviously, this is an extremely labor-intensive and highly open procedure that is not ideal. So we looked around at a number of automated systems, and for cell culture and processing, there are a number of these, and many of you will be familiar with the Prodigy system from Miltenyi, which is used extensively for the production of CAR T cells. But there are other systems like the Wave bioreactor, which has been around for a while, but new ones like the Cocoon from Lovo, the Quantum system, the cell host system, and the Nance system, which are now coming on the market. So we're seeing commercial manufacturers becoming interested in developing devices for cell therapy, cell culture. The automated systems, well, they have a number of pros. They are usually closed systems, so the number of open procedures is extremely reduced. They potentially save time and save labor. They provide fairly reproducible conditions for growing the cells, so one run is pretty similar to the next. They often have built-in monitoring systems and are designed to be GMP compliant. So they provide you with the kinds of documentation during manufacturing that is needed to be in compliance with GMP regulations. And in certain circumstances, there may be no need for a clean room. You could do this in an unclassified space or a class 100,000 space and still feel confident that the cells were protected from the environment. The cons are that the devices themselves are expensive to purchase, usually over $100,000. The disposables that you use with each of the sets, again, they're fairly expensive. And if you have one device, you can only make one product at a time. So this limits your production unless you have the ability to invest in multiple devices. In some cases, it's difficult to monitor the cells directly, so you can't look at the cells while they're in culture. You maybe have difficulty getting test samples to look at the progress of production. And they generally have limited flexibility, particularly those that are computer controlled. There is a limited capability to go into the system and change temperatures or CO2 concentrations and the like. So there is a limited flexibility as a payoff for some of the pros. We chose the quantum system for growing the MSCs. And the major difference is that you can see when we were using T175 flasks, to grow up enough cells for the clinical trial, we calculated that there were over 54,000 open procedures involved in growing the cells in the flasks, whereas with the bioreactors, there was only 133 different open procedures during manufacturing. So a considerable reduce in risk of contamination during cell expansion. Again, the problem is, as you see there, we ended up having to purchase 3 quantum systems and provide the disposables for them, so there was a relative increase in cost. There are other automated systems that can be used for part of the manufacturing procedure. For example, there are a number of cell washing devices. Dave mentioned the Lovo, which has the spinning membrane. And then there are a number of cell separation devices which will successfully purify the cell population that you're interested in or remove the cell population that you don't want in the product. And these are much more flexible in that they're only used for part of the procedure, and one device can do many separations or washings in a day. So again, this is something that's beneficial for the development of new cell therapies. As I mentioned, you can use them with multiple products. They're usually functionally closed systems. They're more reproducible than manual systems, as Dave showed you with the Lovo. They're often faster than the manual methods. Some may be approved by the FDA for use, or you can cross-reference a file at the FDA that has been placed there by the manufacturer. There's the initial expense, however, for the purchasing of the device, and these devices aren't particularly cheap either. And there is ongoing expense for the disposables and the calibration and the service of the device on an ongoing basis. So other considerations I'd like to mention during scale-up and producing large numbers of therapeutic cells, there's the issue of the culture medium. And then there are other manipulations that are involved, not just cell expansion, but you may perform genetic modification of the cells, you may activate them, you may co-culture them with antigen-stimulating cells, and then you will have to potentially freeze them for long-term storage. Then there's release and in-processing testing, which has to be considered as part of the manufacturing process, and then manipulation and retesting of some cell populations before administration. And so in many cases, we will take the frozen cell product out, thaw it, and administer it directly to the patient. But in some cases, the cells may need to be manipulated after thawing. They may need to be washed. They may need to be reconcentrated. And that will require additional testing prior to release. If we look at culture medium, obviously the best choice is one that is GMP grade or USP approved or medical grade. If you find that these are unobtainable, and there are a limited number of such media available on the commercial market, you can use other non-approved medium, but with the FDA approval. So you would submit the details of the medium that you intend to use with a copy of the certificate of analysis from the manufacturer showing how it's been tested. And the FDA will often grant you approval to use this, or they may require additional tests prior to the use of a non-GMP grade medium. There are additives used during manufacture. For example, a lot of cell populations are expanded in the presence of cytokines, and you do not want to be infusing these into the recipient, so you have to have verification of removal of these additives before the product is made available for administration. Genetic modification, obviously with the tremendous interest in CAR T cells, there's a huge amount of focus on genetic modification of T cells to make these products. In general, I would say viral vectors are the easiest to use, but they are expensive to make and extremely expensive to test. So other procedures are being evaluated to see if eventually we could replace the use of viral vectors for gene modification. And as Dave has told you, cryopreservation is useful in that you can freeze down the product and do all of the release testing while it's frozen and prior to administration, but it isn't good for all cells. He mentioned MSCs, and I would mention NK cells do not function particularly well immediately after thawing and administration. So that has to be given some consideration. The other one is scaling up, and Dave dealt with this, and I will just add my 2 cents worth. When you're doing the scaling up translational research towards manufacturing the therapeutic material, where do you get a sufficient amount of the starting cells to do the manufacturing scale up? I mean, these are not readily available. Some commercial companies will provide apheresis products. Some will provide small volumes of bone marrow. But in general, it is a problem if you're having to do validations of the manufacturing procedures. If you're making an autologous product from the patient that is suffering from a disease, normal donors are not the best model to use because they do not have the disease which you're trying to treat, and the cells may behave differently if they're obtained from normal donors as to opposed to an autologous donor. One approach that we have used is to make real products, so therapeutic products, with informed consent from the intended recipient, that these are being used, these cells are being expanded as part of a validation study, and if the validation study does not work, they may not be available to treat the patient themselves, but we will, guarantee to take additional cells when the procedure is validated and make a product that does meet the release criteria. So that is one approach that we've used fairly successfully in the past. Regulatory considerations, well, most cell and gene therapies will require an IND, as Dave referred to. This includes a detailed chemistry, manufacturing, and control section. And in that, you will be required to provide the certificates of analysis for the reagents and materials that you intend to use during the manufacturing procedure. You're also going to have to provide an SOP or a detailed description of the manufacturing procedure. You're going to have to provide a draft certificate of analysis showing the release testing that you're going to do on the product prior to administration. And this will include a list of tests for sterility, endotoxin, mycoplasma, and it will depend on the nature of the product what tests the FDA requires. You'll also be required to produce some stability testing information, and that will include, and it may be that they will allow you to delay this and collect it during the phase 1 study, but they will require some information on the stability of the product during both long-term storage, if it's a cryopreserved product, and also, for example, if you thaw the product after administration, they will often require you to provide stability testing on the thawed product so you know how long it is good for if there's a delay before administering it to the patient. So those kinds of things, they like to see some preliminary data on. And in the release testing, you have to describe the release testing that you're going to use and what method you're going to use for this. And this will include some functional assays. Strictly speaking, functional assays that correlate with patient outcome are not required until phase 3 studies, but the FDA does strongly encourage you, even in a phase 1 study, to try and come up with some testing of the functionality of the cells. So, for example, in the case of CAR T cells, it may be a cytotoxicity assay towards a cell line bearing the antigens to which the CAR T cells are directed. For the FDA release testing, they prefer you to use FDA or USP or EU or ICH methods because these are the approved methods and you don't really have to explain in great detail why you chose that method. Other methods can be used, but they should be approved by the regulatory agency at the IND evaluation status, and you should provide some validation that they are equivalent to the approved method. As I mentioned, functionality is not required before phase 3, but is strongly encouraged. And then there are more complex requirements for fresh cells because you're going to harvest the cells and you have very limited time to do testing before they're administered to the patient. So the recommendation is that you do the testing while the cells are still in culture, towards the final stages of the culture procedure. For example, you would do sterility. You would submit a 14-day sterility test while the cells are still in culture. When you harvest them, you would do a gram stain, and you would also set up additional 14-day cultures. If your 14-day cultures come back positive after the product has been administered, then the FDA requires you to have a procedure for notification of the physician and what you're going to do to address the identity of the contaminating organism and its sensitivity to antibiotics. Very useful documents when you're putting together the chemistry, manufacturing, and control section are these two guidances that are published by the FDA. And they're intended for IND reviewers at the FDA, but they describe in tremendous detail exactly what they expect to see in the chemistry, manufacturing, and control application. So there is one for both somatic cell therapies and one for genetically modified cells. So I strongly encourage you to read these. They provide templates essentially to write the CMC section, and if you follow the instructions, you're almost guaranteed to get through the review process without too many problems. Another big issue is, can we reduce manufacturing cost and time? And I'm going to take an example from our own lab, we were making T cells with specificity towards a number of viruses. So they would be killing of cells that contained EBV, CMV, and adenovirus. And this manufacturing process originally required the production of an EBV transformed autologous lymphoblastoid cell line. So this was lymphoblastoid cells that are infected with EBV, so they express the EBV antigen on the surface, and then we would use them to stimulate the T cells. We would then also transfect the T cells with monomer viral vectors, and that's very expensive. For example, if we were using 2 viral vectors, you're probably talking about manufacturing and testing costs of about $500,000. The method that we used required at least 30 days to manufacture cells for each patient, and that resulted in sometimes that the patients were no longer eligible to receive the cells by the time they were manufactured. So this was the method that we were using, and basically you can see it took 30 days. The cells were manufactured in microwell cluster plates, and so a large number of open procedures, and a highly risky and time-consuming procedure was involved in making these. And as I say, probably about 30% of them were no longer of use to the patients because they'd either progressed or were no longer eligible for treatment. The way to reduce cost was to try and get rid of having to make that lymphoblastoid cell line and infecting it with a laboratory strain of EBV. We also needed to try and get rid of the viral vectors, which, as I say, cost about $200,000 apiece. And so the method to do this was to switch to synthesized peptides as a source of antigens. So instead of using lymphoblastoid cells or transfecting the cells to express the target antigens, we basically used synthesized peptides, and these were overlapping peptides of 15 mers. And for you can see for the PEP mixes that we used, we had one that replaced EBV, one that replaced CMV, and one that replaced the adenovirus. We also switched to the G-Rex devices from the microwell cluster plates. And these are basically containers that have at the base a gas-permeable membrane. And so you just fill them up with medium, add the cells sit down on the membrane, they exchange gas extremely well. This resulted in a reduced number of media changes. They were scalable in that one G-Rex device, if you replaced it with one twice the size, you just added twice the volume of medium and twice the number of cells. And these resulted in improved cell yields. So this was a much more efficient method rather than using all of the open procedures involved with the use of cluster plates. And you can see here that within 16 days, we had more than double the number of cells using the G-Rex device as opposed to the 24-well plates. In conclusion, I think I'd like to just touch on future directions. We have new and improved devices and reagents becoming available from commercial manufacturers, and that is to be greatly welcomed. I think we would all feel more comfortable using automated, closed systems wherever possible. The challenge is that, as I said, that these are extremely expensive, and if you're a large volume manufacturer, you need to invest in a number of these devices to make multiple products simultaneously. The other issue is that we see the biotech companies are trending towards not making devices available or reagents available, but towards making the cell therapy products themselves. So this means that it's kind of moving out of the academic or blood banking environment into the biotech environment. And that means potentially that cell processing facilities in the blood bank and academic institutions may end up only providing collection, storage, and administration infusion services for these products They will basically ship out the starting material, receive the incoming final product, and thaw and administer it. This is true for diseases which have a cell therapy where there is a large number of patients, where the market is fairly large for the commercial company. It is likely that therapies which have smaller number of patients or are less commercially attractive will remain in the blood banking and academic cell processing facilities. So that, I think, is likely to be a future direction. So in conclusion, the clinical scale manufacturing does require a new outlook involving changing methods, reagents, materials, and devices. In addition to cell expansion, you must give consideration to other forms of manipulation, and I've talked about activation and co-culture and genetic modification. Many of these questions that you have when you're moving a cell therapy product towards a clinical trial can be answered by the FDA at what's now called an Interact meeting. This used to be called a pre-IND meeting, where you basically have a teleconference with the FDA and outline to them how you propose to manufacture and use the product. And they will give you feedback as to whether they think your approach would meet with their regulatory compliance, or whether you need to consider different ways of doing things. And I strongly encourage everybody to hold Interact meetings with the FDA. It can save you an awful lot of time, and to do it as early in the process as possible. You don't want to be doing large numbers of scale-ups only to find that the method that you've used will not get regulatory approval. So with that, I would like to thank you and turn it over to the organizers, who I thank for the invitation. Thank you.
Perfect. Thank you, Adrian. That was really very informative. We're now going to move on to the question and answer period. So for our first question for the audience, the question is, what part of translating your work from the lab have you found to be the most challenging? Maybe we'll start with you, Dave.
Sure. I guess my answer to that would be a more general answer, and it would be attention to detail on the research lab end. You know, I think there's a tendency in the research lab to establish methods and do things, and maybe not remember how each little intricate detail impacts the process, and maybe then forgetting about some of those details in that tech transfer. Beyond that, I think it can be challenging at times to translate from culture vessel to culture vessel. And then I think a big thing that I alluded to that's challenging is the whole cryopreservation piece. And I think there's been a tendency across the board to freeze cells much like you would freeze hematopoietic progenitor cells or maybe lymphoid cells. And I think each cell type is unique in their response to cryopreservation, and I think we need to focus more on that end for that final process to ensure that we have the most efficacious product for our patients.
Perfect, thank you. And Adrian, same question for you.
Well, I'd agree with the items that Dave mentioned. I think there are two others as well. One is getting the money to do all of this. The granting agencies are becoming a little bit more forgiving in allowing funds in grants for doing the translational work in the GMP to get this up to manufacturing the final product. And I think the other one that I mentioned previously is getting the source of cells to do the scale-ups. We've come up with one suggestion, but there often can be problems getting a large enough number of cells in which to do the validation studies. So those are the two that I would mention specifically.
Okay, perfect, thank you. For our next question, it is, at what point in my research should I start considering regulatory requirements for cell therapies? Maybe I'll pass it back to you, Adrian, to start.
Basically, as early as possible, as soon as you've got an outline of the clinical therapy that is intended. And this will depend on the preclinical data that is available to support the use of the product, potentially some early animal data, and an outline of the clinical trial. But then, before you've even started the scale-up, I think it's good to have the Interact meeting with the FDA, come up with some basic proposals as to how you see manufacturing and testing the product, and then fly them by the FDA to get some feedback as to whether they think they will meet their approval.
Hey, thank you. And Dave, do you have anything to add as well?
Just probably to reiterate that whenever you are considering your cell as a possible cell therapy, that's when you need to consider the regulatory requirements. But the worst case scenario would be that you consider too late and you have to go back and repeat a lot of your experiments. That's just not a good thing. So earlier is better than later. And occasionally we're involved with groups here, whether that's biotech or at the university here, where you kind of question why we're getting involved so early. But I think, again, you err on the side of early.
Thank you. For our next question, in both of your presentations, you touched on, in an ideal situation, a GMP reagent would be available. Would you be able to elaborate further, in the absence of a GMP reagent being available, what process you went through to qualify that reagent necessary for an IND application? Maybe, Adrian, if you could go first, that'd be great.
Okay, sure. So what we would normally do is we would find a GMP reagent that we'd compared our media against, and we would show the FDA that the cells do not grow well in the approved reagent, but grow better in the test reagent that we want to use. We would then get the certificates of analysis from the manufacturer of that test medium and determine whether we thought they had done enough testing on it. If not, for example, they hadn't done endotoxin testing, we would do the endotoxin testing on the media and submit that to the FDA. So we would have a comparison of functionality of an existing medium and the one we want to replace it with. We would have the certificates of analysis of both showing what testing was done, and we would have results of supplementary testing on the test medium to provide to them to show that it could meet the release criteria.
Hey, thank you. And how about you, Dave?
I agree with everything that Adrian has said. I would just add, occasionally, depending on what type of material it is, there may not be like a GMP equivalent, so you may not be able to do that comparison. And in that case, I think demonstrating or having a discussion with the FDA, on that point, and then, having the certificates of analysis, and which typically would include at least, that safety data, which the FDA would be most concerned about at phase 1. Generally, that approach, when we've had to use it, has been successful.
Great, thank you. For our next question, I believe this may be for Dave. It's regarding manufacturing improvements and freezing at passage 1. Do you freeze every single donor? And if so, would this donor provide sufficient cell numbers for an off-the-shelf product? Or if you pool them from multiple donors, how many donors would you be using, and how many donors do you think would be representative of the entire population?
So for this particular point, I guess, for the MSCs, we wouldn't pool donors. And in general, I think the FDA, I think there have been examples where pooling has been allowed, but in general, that's a difficult path to walk with the FDA. But for the MSCs, we generally would get, at least, half a dozen or so P1 vials with, maybe up to 5 million cells per vial. Each vial then would be, so that's single donor, so a half a dozen from the one donor would allow you then, each vial would allow you to move to a P2 run. So you could get 6, in that case, if you have a half a dozen, 6 runs. Generally, our average yield for an MSC run would be in the range of 4 billion cells. So if you have 6 vials, you can get maybe up to 24 billion cells from that 100 mil aliquot of marrow. So it's true, depending on what your dose is. In the case of these lung injury trials, the dose was quite high, 10 million per kilogram. So you end up needing to do several runs, and I think in the phase 2A study, we needed about 3 or 4 donors. And luckily, you can go back to the same donor to try to limit that variable in the clinical trial. Hopefully that answers those questions.
Great, thank you. For our next question, for Adrian, you touched upon closed systems versus open system manufacturing cultures. If a closed system isn't an option for someone, are there any additional risk mitigation steps that you would need to implement to be able to use an open culture system?
I think what the FDA would look for is what is generally called a fill validation, where you do the processing with just media only without cells, and you do testing at various points of the procedure to check for sterility. And if you do that and submit the data to them, they will then have some evidence that you're capable of performing the manufacturing aseptically, and it should no longer be a problem. But yeah, I mean, the encouragement is always to use either completely closed systems or functionally closed systems. But a fill validation will provide some evidence to the FDA that you are capable of processing sterilely.
Perfect, thank you. And we have had a lot of great questions today, but we're running out of time, so this will be our last question. If you did ask a question, but we weren't able to get to it, we'll be sure to follow up with you afterwards. So for our last question, it is, can you use cryopreserved cells immediately after thawing for clinical use, or is it always a must that the cells should be activated again after thawing, for example, passaging again? Maybe, Dave, if I could pass this on to you first.
I'm sorry, was that to me, Monica?
Yes, that'd be great.
Okay. Yeah, I'm sorry. Yeah, no, I mean, we do both depending on the protocol and, the preclinical data. You can, thaw literally at the bedside and infuse without a wash or without even a dilution or certainly a reculture. I think you want to try to limit the reculture, because if you're reculturing after thaw, then you're going to have to repeat lot release testing. And again, like I mentioned, if that's the practice for a given cell type, it's going to limit access to patients. Only, they're only going to be able to go where you can actually perform a reculture at, typically a company or bio, or an academic center that has an advanced lab that can do that.
Okay, maybe we'll just wrap it up there. Again, apologies if we didn't get to your question. We'll be sure to do our best to follow up with you after the webinar. We've watched just 2 really great presentations on considerations for your translational journey. If you yourself are starting to think about or starting to embark on this journey, the Services for Cell Therapy program at STEMCELL is dedicated to working with researchers to provide tailored compliance and regulatory support and custom manufacturing solutions for your ancillary materials through all stages from preclinical to commercialization. So please feel free to reach out to your local representative for more information. I want to thank our audience members for attending today, and especially want to give a big thank you to Dave and Adrian for your time and sharing your knowledge with us today.